Humans have long held a fascination for the human brain. We chart it, we've described it, we've drawn it, we've mapped it. Now, just like the physical maps of our world that have been highly influenced by technology, think Google Maps, think GPS, the same thing is happening for brain mapping, true transformation. So let's take a look at the brain. Most people, when they first look at a fresh human brain, they say it doesn't look like what you're typically looking at when someone shows you a brain. Typically what you're looking at is a fixed brain, it's gray, and this outer layer, this is the vasculature, which is incredible around the human brain. This is the blood vessels. 20% of the oxygen coming from your lungs, 20% of the blood pumped from your heart is servicing this one organ. That's basically, if you hold two fists together, it's just slightly larger than the two fists. Scientists, sort of in the end of the 20th century, learned that they could track blood flow to map non-invasively where activity was going on in the human brain. So, for example, they can see in the back part of the brain, which is just turning around there, there's the cerebellum. That's keeping you upright right now. It's keeping me standing. It's involved in coordinated movement. On the side here, this is the temporal cortex. This is the area where primary auditory processing, so you're hearing my words, you're sending it up into higher language processing centers. Towards the front of the brain is the place in which all of the sort of more complex thought, decision-making, it's the last to mature, sort of late adulthood. This is where all your decision-making processes are going on. It's the place where you're deciding right now you probably aren't going to order the steak for dinner. So if you take a deeper look at the brain, one of the things, if you look at it in cross-section, what you can see is that you can't really see a whole lot of structure there. But there's actually a lot of structure there. It's cells and it's wires all wired together. So about 100 years ago, some scientists invented a stain that would stain cells. And that's shown here. In the very light blue, you can see areas where neuronal cell bodies are being stained. And what you can see is it's very non-uniform. You see a lot more structure there. So the outer part of that brain is the neocortex. It's one sort of continuous processing unit, if you will. But you can also see things underneath there as well. And all of these blank areas are the areas in which the wires are running through. They're probably less cell dense. So there's about 86 billion neurons in our brain. And as you can see, they're very non-uniformly distributed. And how they're distributed really contributes to their underlying function. And of course, as I mentioned before, since we can now start to map brain function, we can start to tie these into the individual cells. So let's take a deeper look. Let's look at neurons. So as I mentioned, there are 86 billion neurons. There are also these smaller cells, as you'll see. These are support cells, astrocytes, glia. And the nerves themselves are the ones who are receiving input. They're storing it. They're processing it. Each neuron is connected via synapses to up to 10,000 other neurons in your brain. And each neuron itself is largely unique. The unique character of both individual neurons and neurons within a collection of the brain are driven by fundamental properties of their underlying biochemistry. These are proteins. They're proteins that are controlling things like ion channel movement. They're controlling who nervous system cells partner up with. And they're controlling basically everything that the nervous system has to do. So if we zoom in to even deeper level, all of those proteins are encoded by our genomes. We each have 23 pairs of chromosomes. We get one from mom, one from dad, and on these chromosomes are roughly 25,000 genes. They're encoded in the DNA, and the nature of a given cell driving its underlying biochemistry is dictated by which of these 25,000 genes are turned on and at what level they're turned on. And so our project is seeking to look at this readout, understanding which of these 25,000 genes is turned on. So in order to undertake such a project, we obviously need brains. So we sent our lab technician out. We were seeking normal human brains. What we actually start with is a medical examiner's office. This is a place where the dead are brought in. We are seeking normal human brains. There's a lot of criteria by which we're selecting these brains. We want to make sure that we have normal human between the ages of 20 to 60. They died a somewhat natural death with no injury to the brain, no history of psychiatric disease, no drugs on board. We do a toxicology workup. And we're very careful about the brains that we do take. We're also selecting for brains in which we can get the tissue, we can get consent to take the tissue within 24 hours of time of death. Because what we're trying to measure, the RNA, which is the readout from our genes, is very labile, and so we have to move very quickly. One side note on the collection of brains, because of the way that we collect and because we're requiring consent, we actually have a lot more male brains than female brains. Males are much more likely to die accidental death in the prime of their life, and men are much more likely to have their significant other spouse give consent than the other way around. So the first thing that we do at the site of collection is we collect what's called an MR. This is magnetic resonance imaging, MRI. It's a standard template by which we're going to hang the rest of this data. So we collect this MR, and you can think of this as our satellite view for our map. The next thing we do is we collect what's called the diffuse and tensor imaging. This maps the large cabling in the brain. And again, you can think of this as almost mapping our interstate highways, if you will. The brain is removed from the skull, and then it's sliced into 1 centimeter slices. And those are frozen solid, and they're shipped to Seattle. And in Seattle, we take these, this is a whole human hemisphere, and we put them into what's basically a glorified meat slicer. There's a blade here that's going to cut across the section of the tissue and transfer it to a microscope slide. We're going to then apply one of those stains to it, and we scan it, and then what we get is our first mapping. So this is where our experts come in and they make basic anatomic assignments. You could consider this state boundaries, if you will, those pretty broad outlines. From this, we're able to then fragment that brain into further pieces, which then we can put on a smaller cryostat. And this is just showing this here. This is frozen tissue, and it's being cut. This is 20 microns thin, so this is about a baby hair's width. And remember, it's frozen, and so you can see here old-fashioned technology of a paintbrush being applied. We take a microscope slide, and we very carefully melt onto this slide. This will then go into a robot that's going to apply one of those stains to it. Okay. And our anatomists are going to go in and take a deeper look at this. So again, this is what they can see under the microscope. You can see collections and configurations of large and small cells in clusters in various places. And from their expertise, they understand where to make these assignments, and they can make basically what's a reference atlas. This is a more detailed map. Our scientists then use this to go back to another piece of that tissue and do what's called laser scanning microdissection. So the technician takes the instructions, they scribe along a place there, and then the laser actually cuts. You can see the blue dot there cutting, and that tissue falls off. You can see on the microscope slide here, that's what's happening in real time. There's a container underneath that's collecting that tissue. We take that tissue, we purify the RNA out of it using some basic technology, and then we put a fluorescent tag on it. We take that tagged material and we put it onto something called a microarray. Now, this may look like a bunch of dots to you, but each one of these individual dots is actually a unique piece of the human genome that we spotted down on glass. This has roughly 60,000 elements on it, so we repeatedly measure various genes of the 25,000 genes in the genome. And when we take a sample and we hybridize it to it, we get a unique fingerprint, if you will, quantitatively of what genes are turned on in that sample. Now we do this over and over again. This process for any given brain, we're taking over 1,000 samples for each brain. This area shown here is an area called the hippocampus. It's involved in learning and memory, and it contributes to about 70 samples of those 1,000 samples. So each sample gives us about 50,000 data points with repeat measurements, 1,000 samples. So roughly we have 50 million data points for a given human brain. We've done, right now, 2 human brains worth of data. We've put all of that together into one thing, and I'll show you what that synthesis looks like. It's basically a large data set of information that's all freely available to any scientist around the world. They don't even have to log in to come use this tool, mine this data, find interesting things out with this. So here's the modalities that we put together. You'll start to recognize these things from what we've collected before. Here's the MR. It provides the framework. There's an operator side on the right that allows you to turn. It allows you to zoom in. It allows you to highlight individual structures. But most importantly, we're now mapping into this anatomic framework, which is a common framework for people to understand, where genes are turned on. So the red levels are where a gene is turned on to a great degree. Green is the sort of cool areas where it's not turned on. And each gene gives us a fingerprint. And remember that we've assayed all the 25,000 genes in the genome and have all of that data available. So what can scientists learn about this data? And we're just starting to look at this data ourselves. There's some basic things that you would want to understand. Two great examples are drugs, Prozac and Wellbutrin. These are commonly prescribed antidepressants. Now remember, we're assaying genes. Genes send the instructions to make proteins. Proteins are targets for drugs. So drugs bind to proteins and either turn them off, etc. So if you want to understand the action of drugs, you want to understand how they're acting in the ways you want them to, and also in the ways you don't want them to, in the side effect profile, etc. You want to see where those genes are turned on. And for the first time, we can actually do that. We can do that in multiple individuals since we've assayed two. So now we can look throughout the brain, we can see this unique fingerprint, and we get confirmation. We get confirmation that indeed the gene is turned on for something like Prozac in serotonergic structures, things that are already known to be affected. But we get to see the whole thing. We also get to see areas that no one has ever looked at before, and we see these genes turned on there. Is this interesting side effects? It could be. One other thing you can do with such a thing is you can, because it's a pattern matching exercise, because there's a unique fingerprint, we can actually scan through the entire genome and find other proteins that show a similar fingerprint. So if you're in drug discovery, for example, you can go through an entire listing of what the genome has on offer to find perhaps better drug targets and optimize. Most of you are probably familiar with genome-wide association studies in the form of people covering in the news saying scientists have recently discovered the gene or genes which affect X. And so these kinds of studies are routinely published by scientists, and they're great. They analyze large populations, they look at their entire genomes, and they try to find hotspots of activity that are linked causally to genes. But what you get out of such an exercise is simply a list of genes. It tells you the what, but it doesn't tell you the where. And so it's very important for those researchers that we've created this resource. Now they can come in and they can start to get clues about activity. They can start to look at common pathways, other things that they simply haven't been able to do before. So I think this audience in particular can understand the importance of individuality. And I think every human, we all have different genetic backgrounds. We all have lived separate lives. But the fact is, our genomes are greater than 99% similar. We're very, very similar at the genetic level. And what we're finding is actually, even at the brain biochemical level, we are quite similar. And so this shows it's not 99%, but it's roughly 90% correspondence at a reasonable cutoff. So everything in the cloud is sort of roughly correlated. And then we find some outliers, some things that lie beyond the cloud. And those genes are interesting, but they're very subtle. So I think it's just an important message to take home today that even though we celebrate all of our differences, we are quite similar even at the brain level. And what do those differences look like? This is an example of a study that we did to follow up and see what exactly those differences were. And they're quite subtle. These are things where genes are turned on in an individual cell type. These are two genes that we found that as good examples. One is called relin. It's involved in early developmental cues. DISC1 is a gene that's deleted in schizophrenia. These aren't schizophrenic individuals, but they do show some population variation. And so what you're looking at here in donor 1 and donor 4, which are the exceptions to the other two, that genes are being turned on in a very specific subset of cells. It's this dark purple precipitate within the cell that's telling us a gene is turned on there. Whether or not that's due to the individual's genetic background or their experiences, we don't know. Those kinds of studies require much larger populations. So I'm going to leave you with a final note about the complexity of the brain and how much more we have to go. I think these resources are incredibly valuable. They give researchers a handle on where to go, but we've only looked at a handful of individuals at this point. We're certainly going to be looking at more. I'll just close by saying that the tools are there, and this is truly an unexplored, undiscovered continent. This is the new frontier, if you will. And so for those who are undaunted but humbled by the complexity of the brain, the future awaits. Thanks.